Our next speaker, Dr. DeBethizy. Dr. DeBethizy co-founded TEDxWakeForestU in 1997 and serves as president and chief executive officer and a member of its board of directors. Dr. DeBethizy is active in biotechnological industry organizations, say that 3 times fast, serving on its board of directors as a member of both the governing boards of its emerging companies and health sections. Thank you very much. Please welcome Dr. Don.
Thank you very much. Pleasure to be here.
Thank you.
Well, good morning, everyone. This is exciting. It's really great to follow Tony's talk. It's a real live example of the future of medicine. It's a very exciting time to be in medicine. TEDxWakeForestU was founded almost 12 years ago. We actually spun it out of a tobacco company with all of their technology, and you'll get a little appreciation of why we're so excited about this. I'm going to give you an example. We are a public company, so I actually have a little slide in here. I'm going to focus on these neuronal nicotinic receptors, which are located throughout the body, very highly conserved in nature. They're in every organism. Every breath, every thought, every movement of yours is regulated by a nicotinic receptor. That's how fundamental they are. But they had some challenges and people ignored them for a long time. But I'm going to talk a little bit about it. And with the first slide, I am a public company, and since I'm the CEO, I have to be very careful, and you have to be very careful about forward-looking statements I might make. And so just keep that in mind as I go forward. Now, what I want to do is just talk a little bit about how in the world did we get into this. And really, if you look up in the upper left-hand corner, there's a small picture of a tobacco plant. And what you'll find in pharmacology and in most drugs originated somehow from plants and plant-based medicine. So, for example, aspirin came from willow bark. And now we know, and of course nicotine is in tobacco, and people a long time ago in this part of the world discovered that it had meaning to them. They ritualized that meaning, and then later it got developed into modern tobacco products, and then the whole realization about death and disease associated with the product. But fundamental to that, the use of that product, is a compound called nicotine, and it's shown up there with a space filling model. And what that did, as people started to think about what was so compelling about tobacco, and then starting to think about how to treat disease, there were a whole number of reported effects. Schizophrenics are known to self-medicate with tobacco. About 80% of schizophrenics are heavy users of tobacco, and I'll tell you more about that. But actually, one of the things that helped this field the most was the advent of the nicotine patch in the early 90s. By that going on the market, it really became a tool because they developed a placebo patch along with the nicotine patch. And what was exciting about that is people were actually able to do studies. And you can see here that when acute nicotine was given, or the nicotine patch, you could see statistical improvements in learning and memory associated with nicotine. So it started to explain one of the reasons that tobacco was compelling to people, and it was this thing tobacco users would call focus. The problem with nicotine is people started demonstrating activity in a variety of diseases and in patients, small proof of principle trials, but always in non-smokers you saw increases in heart rate and blood pressure. Nausea was really the most limiting thing about the use of nicotine in any sort of clinical setting. And then subsequently, the whole discovery of nicotine addiction really limited the utility of this receptor system, and people were reluctant to move forward with it. Well, we were in the right place at the right time as the genetics revolution came forward, and people began to realize that these receptors were ubiquitous in the body, and they were very diverse. And they're located, of course, in the brain, and it's driving some of the behavior that I just described. And what I wanted to do is just focus on this slide, this picture right here. One of the things, I want you to go back to biology and chemistry now, because I think even liberal arts majors had to take biology at some point. And if you remember, one thing about the nervous system is it's like the nervous system cells connect to one another like wires in a circuit. So you have a nerve cell body, you have an axon, remember the axon, that's the wire, and then it connects to another nerve through a gap called the synapse. Now, there's a whole connection in the brain of these things, and they run all kinds of circuits. They have different neurotransmitters. But what you may not know is that there are interneurons. So this is actually an interneuron coming back on the end of an axon. So here's a nerve cell coming down here. Here's the axon. It's connecting to the next nerve cell that goes on down to another axon. This is an interneuron that basically regulates the activity of that synapse. It's like a thermostat to this room. You get a lot of people in this room, it's going to heat up, and it's actually going to turn the heat off coming from the heater. That's the thermostatic regulation. The nervous system regulates itself that way. The nicotinic receptors in the brain are actually primarily presynaptic, and their job is to turn a synapse down or up. And it was that activity, the opportunity to go after sort of nature's way to correct a deficient synapse or to lower the activity of a hyperactive synapse, that really caused us to go after the receptors. These are ligand-gated ion channels. They're located right here in the membrane. So when that electrical signal comes down the axon, it can be modulated. It can either be enhanced, so you get more neurotransmitter coming out, or diminish, so you have less neurotransmitter coming out. And the great thing about this is that in the early 90s with the molecular biology revolution, people realized that there were 17 genes coding for 17 proteins that assembled in this receptor here with 5 different proteins that cause this ion channel. So here's the electrical signal coming down. Sodium can go into this cell and change that electrical potential and then change the release of the neurotransmitter out the end of that axon. And what we found is every neurotransmitter in the brain was regulated by this system. Not only that, but growth factors were regulated by this system. So it began to explain these are all the areas where nicotine or a related compound has shown some clinical benefit. Not sufficient, not really druggable, but it really gave us the early clue to then start focusing. So you got 17 genes coding into 5 different proteins in one receptor. There's a lot of combinations that are possible. So we had to get real, we had to get practical. If you know one thing about the earlier talk is you have to start as practical as you can get. So what we did is on the bottom, we recognized that a few subtypes were involved in the side effects that were associated with nicotine. We dreamed that if you could target the top subtypes, which were involved in regulating the behaviors we were interested in, and eliminating the side effects by not targeting there, design brand new chemicals, never before created, that had this enhanced activity at what you wanted to do, that you might be able to treat disease. And that's really as simple as Targecept. We named it Targecept for targeted receptors so that we could really envision what we were going to do. So I'm going to talk about one of these subtypes, the one in the upper right-hand corner. It's called the alpha-7 subtype, and a lot of work done at the University of Colorado by Bob Freeman really pointed us to this subtype in the potential for treating schizophrenia. So the whole idea was if you could get more precise about this, you could really develop precision tools. And one of the problems with all the therapies available to date is they're almost like sledgehammers in the nervous system. They're designed to mimic a neurotransmitter. They flood every synapse with the mimic of that neurotransmitter, and you get a profound amount of side effects, and the efficacy can't be controlled very well. If you could target precisely this presynaptic in different parts of the body, in different parts of the brain, then you might be able to selectively start to treat diseases like schizophrenia, ADHD, and Alzheimer's disease. So let's just talk about schizophrenia. Most of you are aware of it from movies, or you might even know somebody. There are about 4.5 million schizophrenics in the 7 major pharmaceutical markets around the world. And it's a really unmet need. Now, atypical antipsychotics are pretty good at treating the hallucinations. But what's not treated at all is the cognitive deficits, which include focus on executive function, attention, memory, and the negative, so-called negative symptoms. These are the things that really limit schizophrenics from enjoying the kind of independence that each of us have in this room. They have anhedonia. They can't seek pleasure. They have affective flattening. Now, I don't have affective flattening right now, but it's a real problem. If I were schizophrenic and go in for a job interview, even a simple job, I'm not going to come across as being enthusiastic about anything. So it's a real problem. They can't hold jobs down. They can't be in relationships like we can because people are not responding to their energy. They also have avolition, meaning they have no motivation to do things. So it's a real serious unmet need. In fact, the head of the FDA Psychiatry Division is so concerned about this that he's given us a roadmap on the kinds of compounds that we should be developing in industry. And the number one mechanism on top of that list is the alpha-7 nicotinic receptor. So we're in the right place at the right time. And I'm going to describe a little bit about the brain here. So this is how the elements of schizophrenia don't function well. So you have positive symptoms being driven by hyperactivity of dopamine release. You have the cognitive inactivity being driven by not enough glutamate release in the brain, and you have the negative symptoms being driven by not enough dopamine. So you've got too much dopamine in one region of the brain, not enough dopamine in another region of the brain, and this is a problem. If you're giving L-DOPA, which is one of the main treatments, not L-DOPA, I'm sorry, but the atypical antipsychotics, they actually, in this slide here, they actually are antagonists of dopamine. So they're trying to lower the amount of dopamine activity to deal with positive symptoms. But they actually can exacerbate the other symptoms because they're not selective enough. So what we did was we knew that the alpha-7 nicotinic receptor were in pathways that regulated dopamine, and they regulated dopamine differentially in different parts of the brain. So you can see here that this alpha-7 receptor, which is located here, can actually improve the glutamate transmission. So we actually discovered a compound that's very selective. Now, we did this through computational drug design. We did it initially in silico. So we do a lot of our design of molecules in the computer. We dock them to an imaginary receptor binding site on the alpha-7 receptor, and then we generate thousands of these and come up with one. And this compound looked very promising, and it seemed to be doing things for both positive symptoms, negative symptoms, and cognition in the preclinical models. So we're very excited. Now we have to go through a very long and expensive process in order to be able to take a brand new chemical that people have never seen before. Number one, we've got to prove that it's safe. So we start with preclinical models. We also have to prove that it can do things in animal models of the disease we're interested in. And I'm very happy to report on the left side here that this compound looked very promising as a potential therapy. So we actually took it into clinical testing. The first thing you have to do is you have to go into normal, healthy people. And we went into normal, healthy people and found out because of the selectivity of this compound, it had exquisite safety. We went up to grams of material in phase 1, where we only need about 10, 15, 50 milligrams to treat what we believe would be a therapeutic dose. We then took it into phase 2, and I'm going to show you those data, and the compound actually worked. It actually worked in a disease that has no approved therapy. So these are the data. This is 185 patients, divided about half and half between placebo and drug. So we have about 90 patients on drug here. And you can see from this graph right here, this is the improvement in negative symptoms. And you can see that we have a very significant effect compared to placebo. This was incredibly exciting. In fact, I had been preparing investors for the fact that the probability of success of this was so low that I had them focused on other things in the pipeline. And then when we hit this, we were astonished. And then the primary endpoint actually in this trial was cognitive dysfunction in schizophrenics. And we thought that schizophrenics were probably self-medicating with tobacco primarily for cognition, because we also knew from phase 1 that this compound was one of the most potent attentional compounds that had been seen by our phase 1 unit. So it was, we were very curious about this, and we actually see again an effect on cognition, on executive function. This is the Groton maze learning task. It's a computerized test where people have to follow a maze, and then they're asked to remember that path through, the correct path through the maze. And schizophrenics cannot prioritize their thinking very well, and they can't do that very well. And what we're finding is they actually improve, and this improvement compared to current therapies is probably about 100% better than anything in the market today. And as I said, there's nothing approved for negative symptoms in schizophrenia. So now the process here is you have to go into a more advanced stage trial. You've got to really demonstrate that you can replicate this finding. As you can see in any clinical trial, it could happen by chance alone, or it could be that the disease condition is so variable that you may not have a strong enough effect to overcome the variability in the patient population. And we will not be able to advance a compound like this unless we can persuade the thought leaders in the field, physicians, scientists, and then the FDA. So this is a trial designed to do that. It's an advanced phase 2 trial. It's in 456 schizophrenic patients. We put them on 4 weeks of their atypical antipsychotic because we can't assess cognition and negative symptoms if they're hallucinating. So you have to get their hallucinations stable, and then you put them on drug. And one of the things we've done is we've actually increased the dose so that we believe we can get even a larger effect based on the trend in the data from the first trial and the learning. So this will be, and we're also going from 3 months to 6 months now, which is what the regulators would like to see for a treatment like this. So just to summarize, I've given you one example. We're working in Alzheimer's disease. We have 2 compounds in phase 2 in Alzheimer's disease. We have this compound in ADHD, actually. We're doing an ADHD study right now. We also have another related compound to this that's actually showing activity in asthma and type 2 diabetes in preclinical models. So we're working broadly across these diseases of the and disorders of the nervous system. And recently we just reported a non-human primate study in Parkinson's disease and L-DOPA-induced dyskinesia. So we're very excited about the possibility of the receptor system. We're moving forward with this compound in an aggressive way because it really is showing the kind of activity that people had hoped for. A little bit of corroborating evidence on this is the fact that another biotech company up in Boston, it's run by a good friend of mine. I've always said we're in the co-opetition mode, but all of a sudden we might be in the competition mode because they also have an alpha-7 nicotinic drug that just showed activity in cognition and negative symptoms in schizophrenia. So we're all very excited. One of the thought leaders in the area said, the central nervous system disease area just got exciting. We've got a horse race going on. And so my friend and I are talking a little less about the details now and a little bit more about how great it'll be to have 2 compounds in development. And we just might be able to improve the lives and restore some independence to schizophrenic patients. So thank you very much.
Dr. DeBethizy, I have 2 quick questions. First one is, how long did you go to school for to learn all this stuff? And the second question is, my God, I'm having a moment. No, wait a second, I'm not kidding. Oh, and the second question, answer the first question, then I'll ask the second question.
You know, that's the reason we're developing these cognition drugs, actually. And actually, it's interesting, as you age, that recall, even, it's going to get worse, So,
well, my mom already told me that I'm going through menopause.
now, what was the first question? I went to school for a long time. In fact, I didn't really know what I wanted to do, so I just kept going to school. And I believe my 24-year-old son, who's still in college, is repeating my performance. So He's out in the audience. But I did get a PhD, and I did do a postdoctoral fellowship. And it wasn't until later I really realized that I'm very applied and very interested in solving problems, and that was best done over on the industry side. So I had to get some training on the business side to be able to do this, but I did that on the job, actually.
Great. You actually answered my second question, which was what brought you into this field.
Thank you very
So
much.
thank you very much.
Thank you.
Thanks a lot.